Marcelo Bigal, medical doctor. He has a medical degree from the University of São Paulo College of Medicine in São Paulo, Brazil. Residency in neurology from the University of São Paulo in São Paulo, Brazil. Master's degree in science from the University of São Paulo in São Paulo, Brazil. PhD in neuroscience from the University of São Paulo in São Paulo, Brazil. Fellowship in headaches from the New England Center for Headache in the USA. President and CEO at the Ventus Therapeutics.
Hi, hello everybody. My name is Marcelo Bigal. I am a neurologist from Ribeirão Preto, from the University of São Paulo, with PhD in neurosciences, and I live in the United States since 2001, and I am the president and CEO of Ventus Biotherapeutics.
Okay, thanks. And what's going to be the theme of your lecture?
I'm going to go over the process of how drugs are discovered and developed. It came to my attention that 25% of the medical students eventually end working for the pharmaceutical industry or for the regulatory bodies, and they have no training on the process of drug discovery. So that's the scope of the presentation.
Okay, so you can share your screen and begin your lecture.
All right. Oops, let's start from the beginning, not from the end. Can you see my screen well?
Yes.
So first and foremost, I want to thank you for this kind invite to be presenting on the science behind drug development. I am particularly grateful from the invitation of my medical school colleague, Dr. Rogério Dutra. And it's a privilege to be here sharing the podium with my former colleague and mentor, Carlos Bordini. The idea of my presentation, I'm going to go over the process, the scientific process of developing medications. And I bet you do not know how medications are developed. And this is a shame because 25% of the medical doctors in the world, they end working for the pharmaceutical industry developing medications, or they end working for the regulatory bodies, such as the FDA or Anvisa, to regulate the process of drug development. And they have to learn everything that's necessary for their work in the process. They don't have any training in the medical school. So I like to basically ask you a very simple question, right? Meaning first, we have to acknowledge that medications are not brought to us by Santa Claus. They require over 10 years and $600 million of investment for every medication that you as future doctors will be prescribing. So the process of starting is actually one of the most complex. Is how do I even start? Which disease I'm going to go after? How am I going to try to modify this disease? How am I going to select chemical matter? How I'm going to optimize this chemical matter? How I'm going to test this chemical matter in animals before even bringing this to humans? So over the course of my presentation, I'm going to go over these several steps with you, taking as an example one medication that I developed all the way through for migraine, and this medication targets this peptide called CGRP, and is a medication that is approved worldwide, including in Brazil under the name Ajovy. So we're going to use this medication to exemplify the late stages of clinical development. But for the early stages of clinical development, I would like to share with you what we are currently doing. So save the migraine target. I'm just introducing the target to you. But now let's start asking the question that I just asked you. How do we even start? I am currently very interested in the field of innate immunity. You know that actually we have two forms of immunity. Innate is born with us, and acquired is developed after an insult or a vaccine. Innate immunity is the first wave of inflammation that follows a lot of diseases, is behind the cytokine storms that happens in COVID infections and other viral infections. And it's behind diseases such as lupus, osteoarthritis, Parkinson's disease. So I'm very interested in innate immunity. So how do I even start? I believe that innate immunity is important. So that's the start. But innate immunity is vast. So basically you have to, at some point, define what exactly you are trying to modify. So starting, I want you to pay attention on how do you believe, guys, that in our cells become healthy, continue to be healthy. Inside our cells, our immunological cells, we have sensors of danger. I am particularly interested in one sensor of danger called cGAS. cGAS is in the cytoplasm. And what is the danger that cGAS senses? DNA. DNA should not be in the cytoplasm. If DNA is in the cytoplasm, there is an infection, the mitochondria is leaking, or the nucleus is leaking, correct? So DNA inside in the cytoplasm is wrong. When cGAS senses DNA, it forms a byproduct called cGAM. Save this because I'm going to show you why this matters later. But the fundamental thing is it causes a release of interferon. So interferon is released from the cells, and interferons is released to try to kill something, right? That very often, but very often there is nothing to be killed. So why did I get interested in cGAS? Because cGAS is behind a lot of lesions that I find, 3 diseases that I find very interesting. So here you're finding dermatomyositis. Here you have cutaneous lupus. Here you have Down syndrome. Actually, just as a curiosity, the gene that codes cGAS is in chromosome 21. So when you have the triplication, right, the extra copy of the chromosome 21 means extra production of cGAS, extra production of interferon, which in Down syndrome cause these skin lesions and systemic lesions. But it turns out actually cutaneous lupus also has facial lesions and systemic lesions that are caused by interferon. If you biopsy these lesions, you can show an interferon DNA signature. And dermatomyositis also have lesions that are very similar and interferon mediated. They have several other similarities. Dermatomyositis has muscle weakness, lupus has muscle fatigue and myopathy, Down has muscle hypotonia. Dermatomyositis has cardiomyopathy, lupus has myocarditis, Down syndrome has cardiomyopathy. So how do you even start? You start by identifying A, diseases that matter and that require treatment. B, a target that is behind these diseases. In my case, cGAS, because I believe that interferons are causing all of this. But then the fundamental question is, right, it looks great, right? Wow, I want to do it. But I know that I'm going to be dedicating 8 to 10 years of my life, and I'm going to be spending $600 million on this development. So the next step is, how do we validate the target? How can I now reassure myself that I am on the right track? So validation sometimes is done by academia, right? The publications from academic centers. Sometimes it's done by experiments. And very often, in the case of experiments, you do genetic modifications in animals. And you try to basically replicate. You try to modify the protein, in my case, cGAS, by a genetic modification and see if the animals develop a phenotype that is similar to what you want to see. And that your medication that you're developing in the chemical lab can modify the phenotype. So that's exactly what we did. We knock it out. We basically knock it out one gene that stops cGAS. So we released cGAS. right? So these animals here, this is called the TREX, which is the model, 1, knockout, minus, knockout. It doesn't have the gene. For SIGS, you do the genetic modification, you raise the colonies, and let me show you how the animal looks like after 8 weeks. See, the animal is sick. The animal is small. The animal is ill. The animal is not exploring the cage, right? The animal is facing an enormous interferonopathy. With that in mind, so now we do the studies. So basically, remember that I told you after SIGAS is activated, SIGAMP is produced, and I said, save this. We now go to the animal here is in the spleen, and we measure levels of SIGAMP. In normal animals, so the level of SIGAMPs are low. In normal animals treated with my medication, it was low, continues to be low. In the mutant animals, very high. After I treat with my medication, 72% decrease. Wow, I did modulate SIGAMP, right? So I did modulate SIGAMP. Then there are other biomarkers that we look. So well, let me keep exploring further. What happens with the animals if we don't do anything with them? So this, you have here the red graphic shows the survival of animals that are not treated. They die. By 63 days, more than 50% of them died. And when we can only start dosing them at the age of 14 days. And after we started dosing them with the medication, we only lost one animal. They survived. Right? So an incredibly survival. So, okay, I modulated interferon, you just saw. The animals survived. They're surviving quite well. And they don't look ill anymore. And now they look normal. They have fur, they're big, they're exploring the cage. Right? They are indistinguishable from an animal that was a wild animal, an untreated animal. So I'm showing what I did with my experiment. That's how we do for any medication, right? At some point, we need to convince ourselves that we are in the right track. So basically, I did that, right? But now the fun really starts. Because how does a target like SIGAS look in real life, right? So let me get to you and show the proteins inside the cell. They look like this. Sorry, let me, I don't know if you're, let me turn off the video. The proteins move inside the cell. And I'm gonna tell you right now why they're moving. And they move and assume infinite conformations, right? Because they're moving, infinite conformations. So now we need to develop an algorithm that cluster these conformations into meaningful groups like 4, 10, 12, 14, so I can start working with. And the reason is, if I don't do that, if I just assume that I average the conformations, in reality, I'm working with something that doesn't exist. What is the average? So if I have 0, and if I have 100, I have two numbers. One is 0, one is 100. The average is 50, but 50 doesn't exist. The true numbers are 0 and 100. So an incredible problem that we had to solve for SIGAS is to identify how many conformations the protein assumes, because what you really want to do is to identify pockets in the protein. If you don't have a pocket, you cannot dock a molecule. You cannot dock a drug. And if you don't know exactly how many conformations a protein assumes, you don't know where the pocket is. You're working on artificial conformation. But you can only, so this is how things happen inside the cell. But in reality, the question that I ask you, do you know why the proteins are moving? The proteins are moving not because the electrical forces are moving the protein. The proteins are moving because they are in water. Cell is water, and water moves. So water squishes and squeezes the protein. Here you're seeing a representation of water. And water will basically is water that ultimately defines the shape of a protein inside a cell, right? Only when you account for that, you can see the pocket of the molecule that actually you now want to drug. So I'm giving you an example now. Then you have to grow the crystals of this pocket. So I'm giving you an example for C-gas, just so that you know. The protein assumes 24 conformations. So the protein goes like, think about a Pac-Man. You're too young to know what a Pac-Man is, but a Pac-Man opening and closing the mouth and flipping, right? A protein is like that. And in the case of the protein of interest, of the 24 conformations, only 4 had pockets. Meaning, if I was averaging them, I would not see a pocket. I would consider this target undruggable and would move away. The target is not undruggable. Actually, the right conformations have pocket. And one thing you need to know about proteins is they will go over all conformations, right? 1, 2, 3, 4, 10, 12, 23, 24, 1, 2, 3, 4. They keep doing the circle. If you stop one conformation, you stop everything that follows and everything that comes before. So you break the cycle, right? So you only need one conformation with a pocket. In this case, we found. Okay, so now I know that the target matters. I know that I am biologically validated. I know that the protein is a pocket. I develop now chemical matter in a laboratory like this. And now I need to find if I bind the pocket. Did I get there? Right? Meaning, did I get there? And the way we try to get there is we try to mimic the structure of water in the pocket. Right? Water is very dynamic. Parts of water are donating bonds. Part of water is receiving bonds. Parts of water behave as oil. So if you develop a chemical matter that has the same properties, we know that water got there. We will get there. We just use water as the blueprint. But now we need to confirm that we do. And we need to define the crystal structure of our protein at the atomic level. Here, what you're seeing is something that we did in our laboratory. It's not C-gas. It's a target called NLRP3, where we identified the structure at a 2.6 angstrom level. Just to give you a sense, a hydrogen atom is 1 angstrom. What you're seeing here is the atomic resolution of the target. And then you are able, when you have this level of resolution, to see if your chemical matter bound to the place where you thought it was going to bind. Now you're seeing, using high resolution electronic microscopes and crystallographies and cryo. We use here cryo-electron microscopy. Then you can see, okay, now I am in good shape, right? The target matters, it's biologically validated. I found the pocket. I developed chemical matter. My chemical matter binds. Right? That's not how the last generation developed drugs, by the way. This is the very modern way of developing drugs. So now that we did all of this, there's just a tiny problem. Typically, our initial chemical matter sucks. It is very, very bad. It binds weakly. Meaning it has horrible properties. So that's the stage of drug development where we call optimization, right? So now that we know that the chemical matter binds there, we optimize the chemical matter, right? We try to make sure that if I am planning an oral, it doesn't get destroyed by acid, right? That basically is absorbed. Tiny little thing, it has to be absorbed, right? That it's not cleared by the kidney in minutes, that it survives after passing through the liver. This process is what we call the optimization of the chemical matter, right? And just so that to know how optimization looks like. So for example, what you see here, Van 5 757. Ven is from my company, Ventus. So this was the first chemical matter that we bound to a specific pocket. And this is the potency against this receptor, the TYK2 receptor. This receptor is, this is not Cgas, I'm just giving you another example. But this is important in multiple sclerosis and several forms of cancer. So it was not potent at all. I had to improve the potency 130 times to become acceptable, right? Something like this, you would have to take a kilo of the medication. Here is how you get to the 10 milligrams dose, 50 milligrams dose, right? In addition to bind to this receptor, I didn't want the molecule to bind to these other receptors. I want that, but not that, right? And it was binding. I had to optimize a lot to show that now I bind here 1,300 more potently than on the other, right? So this is specific. So this is all examples. We spend quite some time optimizing the molecule to achieve, and we generate, see, each of these is one compound. We generate thousands of compounds. But eventually we optimize. And now it's going to be to the next stage of drug development. So if we don't optimize, we failed. There's nothing to advance, right? And actually, as you guys know, we failed more than we succeed. But I just want to now make a pause, because everything that I'm telling you is applicable to small molecules, like everything that you prescribe, Lipitor, Viagra, antibiotics, all of these are small molecules, right? That's how we develop all of them. But there are large molecules, the vaccines and antibodies, they are developed differently. They are not developed like this. And I just want to spend some time with you because I think it's very cool, right? So this is a small molecule, very small, 76 Daltons, 900 Daltons, right? These diabetes medications that you use or hypertension medications. This is insulin, a biological, 6,000 Daltons. This is a monoclonal antibody like the one that I developed at Teva for migraine. Ginormous, very, very big. So for total different indications, right? So monoclonal antibodies are now, meaning the drug of choice for many types of cancers, many types of rheumatological diseases, immunotherapies, and things like that. Everything that I told you, the first part is the same. It's small molecules. You don't need to do all that initial work that I told you because they are bigger than the target. In a small molecule, you have to find a pocket. The large molecule, like an antibody, is bigger than the protein. It doesn't matter where it hits, right? It has many limitations, but it has this advantage. This advantage, which is you don't need to do. But the way we develop antibodies is very, very different. In antibodies, you don't develop in a laboratory like this. The laboratory is the cell. So basically what we do is, so let's say, for example, if I wanted to remember that target that I told you about migraine, CGRP, the way we develop it is we sensitize the rat against CGRP. Rats don't have human CGRPs. So if I inject, rats have rat CGRP. So if I inject human CGRP in the animal will make anti-CGRP antibodies. So now if I replicate the cell, I get a lot of antibodies, but guess what? Cells replicate very slowly. So we now hybridize the cell of the rat with cancer cells. Because what cancer cells do is they replicate very fast. So then, but now they have the genes from the rat. So they are now producing a lot of antibody. But now I cannot use this antibody on you because this is a rat antibody. It's going to cause you a reaction. So I need to humanize the antibody. And the way I humanize the antibody is I change the gene of the rat to not create rat antibody, but to create human antibody. And then I hybridize. And then the cells will produce in very large gallons. Maybe later I can show you how a gallon looks like of cells that could be yeast, that could be mammal cells that will generate the antibodies. But after that, so I just wanted to give you this sense that actually over your life as physicians, 85% to 90% of what you prescribe is small molecule. The rest is large molecule, and that's it. That's what you're going to be prescribing. And that's how they are developed. After this, the process becomes similar again. And either way, we are going to do what's called DMPK studies: distribution, metabolism, and pharmacokinetics. Until now, I want to develop medications I study how my medication modifies the protein. Now, with these studies, I want to know how the body modifies my medication. How is it absorbed? Is it distributed? What is the metabolism? What is the clearance? Kidney, liver. What about in kidney impairment? We have to do, right? That's how we know about dose adjustments and first pass. Does it compete with some cytochromes that are important for other medications? So am I going to have drug and drug interactions? Actually, does the drug get to the tissue? Right? Meaning, if I want to modify the skin, I have to get to the skin. If I want to modify the brain, I have to get to the brain. Right? So that's the next step. And after that is the final step before human studies. It's toxicology. Right? Toxicology is basically trying to predict how comfortable I am to bring this medication into humans, and how cautious or adventurous I can be. We always use multiple species in toxicology. And obviously, the higher the species, the more predictive they are from of humans. Obviously, we do this as very ethically, and we do this very respectful to the animals, but there's no way of not doing this. People very often say, oh, there are ways to test medications without the use of animals. No, there's not. There will be one day, right? When the data sets computer simulations are ready. So at this point, I want to get back to you in transition from my innate immunity discussion to the migraine development, because I'm going to show you how we do this, right? And the reason I'm transitioning is because my innate immunity development didn't get to this point yet, right? But the migraine development got to this point. So how do we do this? Why do we have to? I told you that actually I wanted to inhibit CGRP because CGRP is a powerful peptide of relevance to migraine. But CGRP is a potent vasodilator. So the first question that I have is, if I give CGRP to one of you, am I going to cause a CGRP inhibitor? Am I going to cause vasoconstriction? Are you going to have a heart attack? Are you going to have a hypertension or a stroke because I'm constricting your blood vessels? So that's the toxicology, right? So basically, what did we do? We basically did I'm sure there are many studies that we did, but this one study I find particularly cool. This is a study that we did in monkeys. We did two studies. One was 14 days long, and one was 14 weeks long, because bringing a drug to human is a great responsibility. And the animals were free. They were under telemetry. So monkeys with a holter, a map, continuously monitoring their blood pressure, right? The EKG and holter, and that's it. Just be happy. Eat, jump, run. And we measured this continuously for 14 days. Some of the monkeys got our medications in multiple doses. We gave placebo, 10 milligrams per kilo, 100, and 300. Very high doses. And we demonstrated that we didn't cause any vasoconstriction, any heart issue, anything else. The animals were perfectly healthy, right? So we did a lot of this. Only when we get into this point is now the regulatory agencies, which I'm going to talk a little bit later, give us the go to test in humans. And the testing in humans happened in 3 stages. Phase 1, healthy volunteers. Right? You don't want to test the drug for the first time in sick people. Sick people are sick, right? Phase 2, which can be divided in 2A and 2B, are now diseased people, and it's called the proof of concept, phase 2A, and the multiple dose, phase 2B, the dose response. And phase 3 is the unequivocal proof, confirmatory, at least two studies that find the same thing. That's what clinical development means, phase 1, phase 2, and phase 3. So back to my migraine drug. The therapeutic dose of the migraine drug is 200 in the market. Now we know it's 225 milligrams, right? We start 0.2 milligrams. And then you start raising, right? Then you go to 5, and then you go to 25, and then you go to 100. You know by the animal studies how far you can go. And you know at this point what should be your effective dose. You have an assumption, right? But eventually, we kept doing this until the point that actually we got over, in our higher study, we went to 2,000 milligrams, right? So we started at 0.2, and we went all the way to 2,000. Phase 1 is not one study. So we did, in our case, like here I'm showing 5, but we did 6 studies in phase 1. One was only in the elderly, one was with like very small doses, the one that I just showed you, 0.2, 1. Then we keep going higher. Then we keep testing only man, only woman, the elderly. And basically, this is what we found, that actually basically it was very tolerable, right? Meaning this is as good as it gets, right? Almost identical to placebo in multiple doses, no serious adverse events. Nobody had anything. Most of the adverse events were actually pain at the site of the injection. Right? It hurts a little bit. So when you get to this point, now you are authorized to do late stage development. And that's the point. Until now, you don't need to be a doctor. A PhD, a biologist, a pharmacologist could do this. But this is the moment where doctors add a lot of it. Because that's the moment where you come and say, I'm going to do a late stage development on patients that really deserve this therapy. They matter. Not any patient. The patient that in real life would benefit from a medication like this. For example, a lot of my academic work, and some of these with Dr. Carlos Bordini, was to identify that migraine is a disease of stages. Some stages, migraine is not disabling. People take an Advil. People take Novalgina. People take an over-the-counter medication, they're okay. But sometimes they progress to more severe forms, and eventually some of them get very, very refractory migraines. So monoclonal antibodies are very expensive to manufacture, which means monoclonal antibodies are expensive to buy. So who needs them? Not these guys. Not the guys that benefit from generics. Not the guys that can benefit from topiramate, from amitriptyline. The guys that fail to everything else. So we decided, we did a clinical trial on migraine that was first ever. First, I treated migraine as a continuum. Until then, people would basically either treat episodic migraine or chronic migraine. In clinical trials prior to ours, patients using other prophylactics were excluded. I included them. I basically said, actually, in reality, my medication should only be used in patients that fail those medications, because those medications are cheap. And they work. So there is no reason. So basically, I allowed some of these patients use medication in excess. Some of them use opioids. They are always excluded. But in reality, aren't these the patients that I want to help? Those that, if after 10 years of my life and all these investments, don't I want to do something that actually really has the potential to change their lives? Clinical studies typically exclude patients that fail to pass treatment. I allowed patients that fail to pass treatment. So basically, the change, that was the doctor speaking. I want to include in the trial the patients that I would give this medication, not the patient that I would prescribe something else, right? And obviously I did a lot of studies to convince me that the medication would be effective on here, but that's a patient of my trial. This is a headache calendar, right? And here you're seeing every day of the month. And this is in red. So this is the morning, this is the afternoon, this is the evening. In red, you are seeing the times where the patients could not leave the bed. In orange, the patient could leave the bed but had very severe headache, couldn't work, couldn't study. And in yellow, the headache was there, but they could force himself or herself to work. What you don't see in this diary is lack of headache. You don't see. Show me one day here where these patients had always been excluded from trials because they're horrendous. They're refractory. I didn't exclude them. I treated migraine as a continuous where remember I told you phase 3, I did 2 phase 2s, not one, and 2 phase 3s. 1 phase 2 in chronic migraine, 1 in episodic migraine. So the patient would come to the same clinic. And if the patient had 15 or more headaches per month, the patient was on this trial. If the patient had less than 15, the patient was on this trial. We never lost the patient. The medication placebo, medication monthly, or medication quarterly, once every 3 months. And then after that, the patients would, and then we did the phase 3 with the exact same design. And after this, all patients would roll to a 1-year follow-up receiving drug, right? So this is 3 months. Then everybody continues to receive drug for 1 year. Now I'm not saying the primary efficacy, I'm looking to safety. So we show that both, this is episodic migraine, both our doses work very well in episodic migraine. Both our doses work very well in chronic migraine. Look to the p-value in chronic migraine. These are the patients that people exclude. And then we showed in phase 3 that basically the medication separated from placebo after 1 week only. And actually, I have a p-value in 1 day. After 1 day of therapy, they had significantly less headaches than placebo. And then it was very fast. It was sustained. It worked in both doses. And we were able to show other endpoints, moderate or severe headaches, number of migraine days, but that's what matters. Look the difference in between this is the patient that I showed you. This is not live. You probably never saw a patient like that, but you have one of the world authorities of headache in your faculty, Dr. Carlos Bordini. He saw this for a living every day. Sit with him. And don't ask Dr. Bordini to teach you about migraine. He will teach you about migraine. Ask him what it is to live with this. And what is the difference between going from this to this? The patients need to relearn how to live. A patient like this, don't go outside because the sun is intolerable. A patient like this needs to learn to go outside, trusting that the sun is not going to burn, right? And so far. So once we have all of this, it's time to go to the FDA, Anvisa, the regulatory bodies. That because regardless of the times that we live, science does matter. Science does matter, right? And the reason we regulate drugs need to be regulated is because in the past, you only need IP, intellectual property, to launch a drug in the market. So people launched cocaine for toothache. People launched sulfonilamide, which not knowing because they didn't do the DMPK studies, that this is converted in the body in alcohol that is poisonous and that killed hundreds of people, mostly children. The mothers were giving this to stop their cough. This was being converted into methanol that was poisoning the child. I don't need to tell you the story of thalidomide, a medication that was developed for nausea and was given, well, pregnant women have nausea. Let's give thalidomide for pregnant women. And you know this story. You know how it ended because the proper development was not done, right? So basically, what the agencies will look is not only your clinical data, they will look to all your non-clinical package, the quality of your manufacturing. I don't know if you're following, but losartan, one of the greatest antihypertensive medication, is being removed in Brazil by Anvisa because of a contamination in the manufacturing process that allowed particles that are cancer, that can cause cancer. So the agencies monitor the quality, the preclinical, the clinical package, the regional needs. So for example, China and Japan required that we do a dedicated study in China, Japan, because Oriental people metabolize drugs differently. So there is a good reason to do that. So they will assess, then we launch, and then we commit the next many years of our life agonizing about following the drug in the marketplace, because at this point, when we launch a drug, we have unequivocal proof that the drug works. We have very good sense about that the drug, the tolerability. But despite the fact that at this point, we tested thousands and thousands of patients, rare side effects are missed in the clinical development. So now only when thousands and thousands and thousands of people use the drug, you are capable of identifying very rare events. And that's the risk, meaning if you ever heard about this medication, Accutane, Roaccutane, which is a medication that actually is used for acne and is incredibly teratogenic. Meaning if a woman becomes pregnant, the child will be born with incredible malformations. And if any of you ever used this medication, you know how the risk mitigation for this is severe, right? Meaning my daughters used, they had to do a pregnancy test at the age of 12 every month before getting the prescription. They had to do liver tests, right? Because that's how we guarantee the safeguard of the population. So let me just finalize to basically say, I hope that I convinced some of you that to consider drug development as a profession, a medical profession, because at the very end, you are going to be giving up on treating patients on a daily basis, but you're going to be treating lots of patients through the prescription by doing an incredibly good work through the prescription of other doctors. And I just want to share with you why we do it. I mean, this is a letter. I'm going to share some of the letters that I got when I was developing. I developed multiple drugs in my life, a medication for Huntington's disease, several drugs for asthma. This migraine drug I developed from, I am the inventor. I am the patent inventor. I developed from the chemical matter. And it's very gratifying when it works, right? So listen to this. Dr. Bigal, I'm a long chronic migraine patient. I've been struggling. I use this. I'm desperate. Can I join your clinical trial? I'm a resident in the United States. I hope this doesn't preclude my participation. Obviously not. The trial was being done in the United States. Stina, it's after midnight. I'm sitting in my living room. This is a mid-20s chronic migrainer. I'm sitting in my living room, scouring the internet from any new information. The last hour, I've been silently crying because I barely made it out of the shower. I'm yet on another new medication in an attempt to prevent my migraines. These are the patients that are excluded from the clinical trials. I have a migraine nearly every day. I saw 5 neurologists, MRIs, trigeminal nerves, nothing with success. Can I try your medication? This is a man, a young man. I feel that I have been robbed of being allowed to live my 20s and my life is pretty much on hold. My head feels like a portable prison on the top of my shoulders. I don't have normality, just pain and noise relentlessly. Sorry if this sounds negative, but a constant headache for 9 years does leave you in despair. That's what I had to tell you. I hope you found this relevant. I tried to cover the process of drug development. We live in horrible times where everybody has an opinion about what it works, what it doesn't work, and everybody thinks that they know better. But in reality, the process of drug development represents 10 years of heavy studies and investment that is incredibly serious. That if we do very well, we stop bad medications from hitting the public. We allow great medications from hitting the public, and people benefit, and the public benefit. I hope I gave you a tour over how new medications are developed. I'm very pleased to see that I did this in 44 minutes and 12 seconds. And I'm going to be joining you on the day of the meeting for a Q&A. Thank you very much.
Thanks, Dr. Marcelo. I think it's very important that you gave this lecture, and we are very thankful. I think it's an inspiration for all of us that a Brazilian doctor is where you are, doing what you're doing, and thank you very much.
My pleasure. Thanks for the invitation. If I can say one more thing before the end, I don't know if this is being recorded or not. If it's being, it's for everybody. If it's not, it's for you. Whoever is in the field of medicine for the wrong reasons, for money, for prestige, whoever is doing this in a hurry should not do it. If you want to be a good doctor, if you want to be a good scientist, if you want to be somebody that actually adds a good teacher, you need to be able to really do this for the right reasons, spend your time, be very patient, and enjoy the profession, right? Nobody forces us to do things in a rush. Nobody forces us to be disrespectful. Nobody forces us to degrade ourselves by accepting jobs that actually are not good quality jobs. So good luck for you in your careers, and I hope you enjoy the meeting.
Hi, Dr. Marcelo.
Hello.
Hello there. Can you hear me? Can you see me?
Yes. How are you?
Doing very well, thank you.
That's great. I'd like to thank you for your lecture again. It was awesome. And now we have some questions for you. Can I start?
Please, please.
So the first one is, the development of a research question begins with the identification of a clinical problem that is important for the patient, related to the management, and finally the improvement of their health. In your opinion, what would be the criteria for developing a good research question?
That's a great question. So good, you mentioned some of the criteria, right? Good research questions matter. They're relevant. Right? They will attend the problem that impacts people. In that regard, I think the most important characteristic of research question is it is precise. Right? So basically, a good research question is very well defined. Right? When Einstein was studying why light was deflected by gravity, the research question was, does gravity deflect light? Yes or no? Does fremonezumab work for migraine? Yes or no? Is the improvement meaningful? Is the tolerability acceptable? So research questions have to be precise. And they have to be precise, they have to be relevant, and we should be able to answer them with an experiment. If you're not capable to answer them with an experiment, they're not research questions. They are philosophical questions or speculation.
Okay, thanks. So the next one is, in your opinion, what's the main difference between scientific research in medicine in Brazil and in other countries? What are the difficulties, advantages, and disadvantages?
I think it's easy to say that research in Brazil is more difficult than elsewhere. And in many aspects, it is, right? Because, for example, when I was a faculty at the Albert Einstein College of Medicine, we were 80 neurologists as professors of neurology. I guess that actually the University of São Paulo is 1/10 of that, like 8, 9, 10, right? So there is this issue of the volume not being the same. There's also the issue of technological access being very different. But then I don't think that Brazil is in the bottom of the scientific chain at all. I think Brazil is a country that is compatible with very good quality of research. And the best advice that I give to you is don't do second tier research. If somebody in the United States is doing a study with PET scan, and their PET scan is wonderful, and here we don't have a wonderful PET scan, don't do a PET scan study. Try to see what we have here that they do not have there. And I'm going to give you a concrete example. When I finished, I was doing my master's, and I was working multiple jobs to survive as a doctor, including in the public health system in Ribeirão Preto. And I would see 100, 150 patients in a given day. And so the question to me was, but that public health system did not have MRIs, did not have any of the fancy stuff. But what did they have? Patients. So what was my first publication in the Headache Journal was a study when I personally assessed 6,006 patients, I still remember the number, and mapped every single headache that any one of them had. Many or most didn't. Some of them had 1, 2, and 3. And this became the largest South America epidemiological study ever done, where all the patients were interviewed by the same doctor. So out of the blue, our disadvantage became our advantage. A second example that I would give to you, when I was in the emergency department training as a physician, how did we treat migraines in the emergency department in Brazil? With dipyron, IV, diclofenac, chlorpromazine, magnesium sulfate, IVs. Guess what? There was never a single study testing these medications because in the United States and in other countries, they don't treat it. So I conducted what was the first randomized clinical study with these medications and published this in multiple papers with Bordini, Speciali, and others, encephalgia. So my recommendation to you is opportunity is everywhere. Don't commiserate that we are disadvantageous. Don't do second-tier research. Go for what we can do that others can do.
Okay. And what are the main challenges faced during your research? Could you describe the difficulties and rewards that have been provided at this relevant investigation?
The main, well, talking about my Brazilian years, the main challenge for a Brazilian investigator, and I think it's still kind of like that, especially for the doctors, is that unless you dedicate yourself fully for the post-graduation, you are not paid. But when we finish our residency, we're 26, 27, we have to work, right? So we eventually end doing our research for free, and it becomes demotivating. It becomes a little bit a disincentive. Not a lot of people are lost because of that. But my main recommendation is what I said at the end of my presentation. Nobody is forcing you to do research. You should only do research if you really like it. There is nothing wrong in not doing research and being a superb clinician, dermatologist, radiologist, or surgeon, right? So, but if you're going to do research, you have to have passion for it. You have the main challenge is research takes time. You're going to invest years of your life. Sometimes you're going to be working to the middle of the night, and very often you are going to finish your research, and you're not going to find anything glamorous. You're not going to find, right? But I think so. The fundamental challenge is, what is my motive? Why am I doing this? Am I doing this because I really like it and I couldn't live elsewhere? Then you become a researcher. Those of us who want to be full-time researchers, we are okay not seeing patients. We are okay at some point, and I love some patients, but we are okay basically making less money than, for example, if we were doing surgery or things like that for most of us. But we don't see ourselves doing anything different. I think that's the point that I would convey to you.
Thank you. The next question is, Dr. Marcelo, in your opinion, what are the impacts of nanomedicine on our health?
Of nanomedicine?
Yes.
I don't think that actually I cannot answer this question yet. I think that actually to answer this question is the same difficulty of answering what's the impact of stem cell research in our life, of RNA interference in our life, of modalities, right? I think there is a very good argument to be made that nanomedicine, both nanomedicine in terms of clinical development, but also nanomedicine in terms of surgery, the microsurgeries that actually will take advantage of nanomedicine. There is a good argument to say that they are promising, but there is still they're new. There is still a lot to be done to generate the evidence that actually we unequivocally answer if they are yet as promising as they seem to be. So I'm sorry if this is a little bit of a frustrating answer, but that's my position.
Okay, next question. What are the main bioethical issues involved that can delay progress in the nanomedicine area and in the development of medicines that would help the population in a great way, like your research?
I think the bioethical issues that involve nanomedicine are the same bioethical issues that involve research, right? So the fundamental question is, meaning, research is only ethical if we reach what's called the equipoise, right? Which basically it's the potential benefit supplement the potential harm, right? So, for example, you don't know if if a medication is going to have benefit until the end, and you don't know if the medication is going to have harm until the end as well. But at any given point, you have to be convinced that the potential for benefit is higher than the potential for harm. So when we start the trials, we have to believe that. As we start seeing side effects, we have to be able to reassess that and discontinue if the equipoise is not reached. So, for example, right now I'm dealing with a dilemma of a medication that I'm developing. It's very new, first in class, and this medication plausibly would be beneficial for people with Down syndrome, syndrome de Down syndrome, right? And this medication also could be potentially beneficial for lupus and dermatomyositis. Oh my gosh, how much I would like to help people with Down syndrome. But am I ethically authorized to start testing people with Down syndrome in the absence of any safety package? I only have animal data so far. Or should I start more modestly and start with lupus, only look to their scan, increase my dose, build my safety package, and go to Down syndrome? So the bioethic dilemma is inherent to clinical research, and it's not different for nanomedicine. So the fundamental question is, for those of you, I mean, because I'm getting these questions, I'm very sure that some of you are interested in the field and are considering that. It doesn't matter. Back to basic. Are you evolved enough in your thought process to bring this to humans? And do you strongly believe that the potential for benefit is higher than the potential for harm? if you can answer this question positively, go for it. For nanomedicine, for stem cell therapy, for small molecule, for vaccines. It's the same rigor.
Okay, last question. I found it very interesting to know that a doctor is of fundamental importance in the development of medications, not only in clinical research, but also during the development and functional tests. What suggestion would you give to those who are interested in building a medical career together with research?
Do it. It's not incompatible, right? So, for example, just listen. Before being a head of research and development, I was for 3 times chief medical officer of very large institutions, top doctor, chief medical officer. And do you know what a chief medical officer does? The chief medical officer is not the voice of the doctor. The chief medical officer is the voice of the patient. So, why do I strongly believe that doctors are great drug developers if they do it well? Because they have this touch with the patient. They understand the disease. I could have never developed my migraine medication so well if for many years I have not practiced as a headache. I knew what the patients need. So if a doctor wants to become a developer, develop from the perspective of the patient. Because those that did not study to become a physician or a nurse cannot represent this voice. Second, if for your own happiness and if your own development skills, practicing is important, do it. Nobody's preventing you from that. Carve your time, 30%, 70, 50, 50, and just make in a way that actually is one fits the other. If you develop drugs, you become a better doctor. If you're a good doctor, you develop better drugs. It's perfectly compatible to do both.
Thank you so much. Dr. Rogério is congratulating you on your lecture. And on behalf of our team, we'd like to thank you for being here and for giving your lecture. I think we all really enjoyed it. And we really look forward to having you back next year.
I'm looking forward for next year, and please give a hug to Dr. Rogério and say hello and the best of luck for you. Great meeting and congratulations.
Thank you.
Thank you.
Bye.
Bye.
Então, ó, padrão clássico de uma meningite bacteriana. Vamos lá, olha. Celularidade.
A sua melhor escolha para residência médica é a Medicel. Sabe por quê?
Primeiro, porque é multimídia, com diversos conteúdos em todos os formatos que cabem na sua rotina.
Questões comentadas, simulados, vídeos, podcasts, textos, livros totalmente impressos. Amiga, tem tudo.
Estudar em qualquer hora, em qualquer lugar, é perfeito para mim.
Tem até um orientador de estudo para me apoiar. Ele traz dicas e até monta um cronograma com foco no que eu preciso estudar.
E a nossa plataforma é inteligente e personalizada. Baseado na prova que você vai prestar, ela sabe exatamente aquilo que você precisa aprender e traz questões e conteúdos comentados dessas provas para você treinar.
E se você sentir que precisa de uma força, a gente está aqui para te orientar, te acompanhar, tirar dúvidas com os professores ou até mesmo montar um plano de estudos para você.
É o melhor dos mundos. Uma tecnologia incrível que a gente acessa de qualquer lugar.
Com aulas fantásticas e professores atuantes que sabem tudo sobre as provas de residência médica.
Com informações e dados para a gente saber exatamente como está nosso desempenho frente aos outros alunos que prestaram as mesmas provas.
Medicel é o melhor jeito de estudar
E de te fazer passar.
Acesse agora
E aproveite 7 dias grátis.
Hello everyone. So I would like to say that we're going to be live in a few moments with Dr. Aaliyah and Dr. Patricia, and we're very excited for this. We have been looking forward to this moment since last year. So we really hope you guys have here and stay here with us.
Fala pessoal, boa tarde. Agora, em poucos minutos, a gente vai ter uma roda de conversa com a doutora Lia, com a doutora Patrícia. Nós vamos ter uma intérprete junto com a gente. A gente está muito animado por esse momento. Ela vai falar sobre medicina de família, sobre SUS e tudo que isso causa e o impacto que tem isso no nosso país, no país dela, tá certo? A gente espera que todos vocês estejam com a gente. Vai ser muito especial esse momento. See you soon.
from other countries, especially when we know that the United States has a
se eu posso mudar quando eu quero para escutar elas falando.
A Letícia perguntou se pode mudar quando ela quer para escutar elas falando.
Interpretation.
Ó, vai no... So we're gonna start. We're live.
Can I start?
Yes.
Hi, I'd like to say thank you for Ali and Patricia. It's a pleasure to have you here. We are so happy for this. I hope you will great the Congress. It's a pleasure.
Thank you for having me. It's a pleasure to be here. Hi. Thank you everyone for this opportunity, and most thank you, Dr. Alia, for being here with us again. So I think we can introduce ourselves. So I'm Patricia. I'm a professor at the university here in Franca, Uni-FACEF, and I'm family physician here and geriatrician too. And
pediatra. E gostaria de saber um pouco mais sobre a senhora, doutora Lia, sua formação, onde a senhora é, como é a sua prática. Então, por favor, a palavra é sua. Fale conosco. Olá, muito obrigado por me receberem, doutora Patrícia, e todos os alunos por me convidarem. É muito emocionante e é maravilhoso que as mídias sociais nos dão essa chance de conectar e aprender uns com os outros de milhas e milhas de distâncias em diferentes países. Eu me tornei médica da família e, deixa eu pensar aqui, bom, deixa eu falar do início. Originalmente eu sou da América do Sul, assim como vocês. Eu me mudei para os Estados Unidos quando eu era adolescente. Eu estudei aqui e depois vou falar com vocês sobre meus estudos, mas eu fiz a universidade aqui, a faculdade de medicina aqui, e eu terminei toda a minha formação em 1999. Então agora eu fiz 50 anos e agora já estou praticando há mais de 20 anos dentro dos Estados Unidos. Então quando eu me formei e terminei minha formação, eu comecei a trabalhar para a Universidade do Sul da Califórnia, lá na Califórnia, eu trabalhei por um ano lá. Depois eu resolvi que era muito difícil, porque a gente estava na Califórnia e era muito longe para dirigir até o trabalho. Então eu aceitei um trabalho na Kaiser, que é uma grande empresa, uma empresa seguradora, onde eles têm seus próprios escritórios, seus próprios hospitais. Então eu trabalhei ali por muitos anos, talvez por 5 anos. Depois eu comecei a trabalhar com... Eu comecei minha família se mudou da Califórnia para Chicago. Então quando eu me mudei, minhas crianças e meus filhos eram muito pequenos. Então aqui nós temos o que a gente chama de tratamento de urgência. Não é uma sala de emergência, mas as mesmas doenças de resfriados, de tosses. Então as pessoas vão ali para se consultarem com o clínico geral normalmente. E depois, mais ou menos 4 anos atrás, eu decidi que eu queria começar meu próprio consultório. Então eu abri um consultório e não atendo pessoas que têm seguro-saúde, então as pessoas têm que pagar para se consultar comigo. Então isso é um pouco diferente de como as coisas são nos Estados Unidos. Então agora eu tenho a minha própria clínica, eu faço tudo sozinha, eu não tenho enfermeiras, eu tenho só uma assistente que não vem todos os dias, ela fica algumas horas durante a semana para fazer, eu gosto de fazer tudo sozinho, e é um novo tipo de tratamento médico que está acontecendo aqui nos Estados Unidos. Na minha região aqui, no meu bairro, tem quase 1 milhão de pessoas, de habitantes. Eu sou a única pessoa que está fazendo isso em todo o estado, em toda essa região. Mas eu gosto muito. Eu acho que nos Estados Unidos, provavelmente, é uma das melhores formas de trabalhar como médica. Então, como vocês podem ver, eu trabalhei em 4 lugares diferentes e eu vi o sistema público americano de diferentes lados e com diferentes práticas familiares e como é bom se tornar uma médica aqui. Então, essa foi a minha história. Muito, muito interessante, porque, pelo que eu sei, o sistema americano de saúde só fala de seguro. Você não tem um sistema universal de saúde como nós temos aqui no Brasil. E como você disse, você trabalhou para uma grande empresa, uma grande seguradora, então eu queria saber um pouco mais sobre esse emprego, esse trabalho, como os pacientes chegam a ter esse tipo de atendimento. Porque aqui no Brasil, nós temos 2 grandes sistemas. Nós temos o SUS, que é o nosso sistema universal de saúde, que é para toda a população, para todos aqui. E também nós temos o tratamento particular, que as pessoas pagam para ser consultados e para receber atendimentos. Então, você pode nos contar um pouco mais sobre essa seguradora que você trabalhou? E depois disso, se a senhora puder dizer também sobre o Obamacare, que eles chamam aqui. Com certeza. Bom, deixa eu explicar primeiro os diferentes tipos de seguro que as pessoas devem ter. Então, você pode ter um seguro que você se qualifica se você ganha até uma certa quantia de dinheiro, a gente chama de Medicaid, que é para todas as pessoas que são pobres, que não podem pagar por um tratamento. Então, eles têm esse Medicaid. E entre esses, quando você se torna mais velho, tem outros seguros, mas você tem que pagar por eles. Então, alguns deles, se você trabalha para essa empresa, a empresa pode pagar parte do seu plano de saúde. Então, se você não trabalha por uma empresa dessa, você tem que pagar você mesmo. E é muito caro, pode ser talvez $1.200 por mês para uma família de 4 pessoas, o que é muito dinheiro aqui. Se você trabalha em uma empresa que pode pagar metade disso, então você pode pagar 600 e a sua empresa paga a outra metade. Então, quando você envelhece, nós temos o seguro do governo para todos os idosos, que a gente chama de Medicare. E todo mundo consegue ter acesso a isso. Mas o negócio dos seguros é que nem sempre paga tudo, por tudo. Costumava ser assim quando eu era criança, ou lá nos anos 70, eles cuidavam de tudo. Você ia ao médico e você colocava o cartão do seguro e você ia para casa e não se preocupava com isso. Agora é totalmente diferente. Mesmo quando você tem o plano de saúde, né, o seguro, às vezes eles dizem, você tem que pagar os primeiros $6.000 e depois disso nós vamos pagar. Então, para muitos pacientes, talvez você pode ir ao médico 2 vezes ao ano, ou 2 ou 3 vezes ao ano, e isso gasta mais ou menos $200. Mas você vai pagar por isso até conseguir pagar 6.000. Espero que tenha feito sentido. Então, para responder a sua pergunta, os seguros, os planos de saúde são muito complicados aqui nos Estados Unidos, mas a grande lição é, infelizmente, não paga por tudo. Então, para os pacientes, você tem que decidir qual é o melhor tipo de seguro, os mais caros ou o que cobre mais coisas. E se você estiver pagando $1.800 por mês, mas quem tem esse dinheiro? Então é melhor pagar talvez 300, mas você não vai a médico com tanta frequência. Ou talvez você tem câncer e você tem que ir ao médico mais de 2 vezes ao ano. Então você tem que pagar 2.000 por mês para ter acesso. Então nós temos uma grande diferença de preços aqui e qualidade também. Então, quando se fala de trabalhar para empresas que são seguradoras ou planos de saúde, isso é um tipo de emprego. Eu lembro que quando a Kaiser, que eu trabalhei, foi meu primeiro emprego depois de sair da formação, era um local muito bom para trabalhar, tinha muitas unidades, era muito perto da minha casa, e tinha muitos outros médicos trabalhando, os médicos jovens, então nós tínhamos muita companhia, você podia perguntar alguma coisa a alguns outro colega, então os colegas se ajudavam, mas eram muito rígidos sobre a forma como nós praticávamos medicina. Então, por exemplo, se alguém tiver uma infecção no nariz, você não podia usar. Aqui nós temos uns medicamentos que são um pouco mais caros, você não podia prescrever esses medicamentos, não pode, você não tem essa permissão. Você tem que usar amoxicilina ou um genérico, você tem que usar isso. Então, e você tem que usar os mais baratos, depois ir subindo. Isso não é tão ruim porque é mais ou menos senso comum. A maioria de nós aqui no tratamento médico, a gente faz essas coisas que sejam acessíveis aos pacientes. Mas outras coisas eram mais difíceis. Por exemplo, você tem um paciente com dor nas costas e você quer que esses pacientes vão a um ortopedista. Eles não podem ir, a não ser que passem por fisioterapia ou ter esteroides ou uma injeção. Então a porta está fechada até que você prescreva essas coisas. Então isso é um pouco mais difícil, porque talvez você pode ter uma opinião, mas você tem que discutir com alguém. Então é uma forma mais controlada de trabalhar. Então naquela época, e acho que isso foi em 1990, ou no início dos anos 2000, na verdade, muitas pessoas não queriam trabalhar lá porque elas diziam: as seguradoras, os planos de saúde querem controlar o que nós fazemos. Então é muito melhor você ter a sua própria clínica, o seu próprio consultório. Então o que aconteceu aqui nos Estados Unidos é que nós tivemos grandes grupos chegando e eles compraram muitas clínicas. Então agora, a maioria dos médicos trabalham para grandes grupos e o benefício disso é que você tem mais acesso aos pacientes, o seu salário está garantido, você pode ganhar bom dinheiro, mas também você passa por essa rigidez. Eles te dizem: você tem que atender essa quantidade de pacientes. Você tem 4 pacientes dentro de 1 hora. Então 20 pacientes ou 30 pacientes em um dia. Então eles vão controlar isso. Então isso se torna mais difícil. Então é por isso que muitos médicos agora estão voltando a trabalhar na forma como eu fiz, que é trabalhar sozinho, de forma independente. Respondi sua pergunta, só para começar? Sim, isso responde muito, na verdade. É bem semelhante e é interessante porque, apesar dos sistemas serem totalmente diferentes, a forma como eles funcionam são bem semelhantes. Porque aqui, quando a gente faz essa gestão da nossa agenda dos pacientes, nós ficamos rígidos também a alguns protocolos na área particular. Então, só alguns exames que eu posso prescrever para os meus pacientes. Outros eu tenho que encaminhar para o outro profissional. Apesar de eu achar que eu sou capaz de fazer aqui ou esse tipo de investigação. É bem semelhante na forma como funciona, apesar de ser diferente na essência dos sistemas. Outra pergunta que eu teria é sobre sua prática agora como uma médica independente. Nós sabemos que é um problema que nós temos aqui no Brasil. Quando você está trabalhando sozinha, você tem uma assistência, você pode fazer a sua avaliação clínica do paciente, mas quando você tem que pedir exames ou trabalhos complementares, como é que esses pacientes podem pagar por esses exames? Porque se eles não têm um plano de saúde para cobrir por isso, que você não é o médico do plano de saúde. Isso faz sentido, minha pergunta? Sim, com certeza. Bom, o que eu quero que todos saibam é que o mundo está mudando. Não há nada perfeito hoje em dia. Pode mudar de novo daqui a 10 anos. Então eu nunca quero ser alguém que faz com que as pessoas se sintam desestimuladas sobre a medicina aqui nos Estados Unidos. Na verdade, pelo contrário. Eu acho que a medicina é a área mais incrível. Nós temos muita sorte, todos nós temos muita sorte, porque a gente cuida das pessoas, a gente consegue cuidar das pessoas. E, claro que existem a parte difícil, pessoas que estão cuidando disso tudo, mas o que é importante é você cuidar do seu paciente. Então, para responder a sua pergunta, na minha prática, eu tenho pacientes que têm plano de saúde e muitos que não têm plano de saúde. Então, na forma como essa prática é montada nos Estados Unidos, é que os médicos têm a sua própria conta com o laboratório, então eles podem encomendar a exames para os pacientes dentro dessa conta. Então, o laboratório vai me cobrar e vai descontar do paciente. Então, em vez de cobrar o paciente, só os pacientes, $100 por que no CBC, eles vão me cobrar $5, por exemplo. Então, eu vou cobrar do cliente e pagar ao laboratório. Então, quando o laboratório se junta à minha prática, é um dos benefícios que eu recebo deles. Então, eles podem fazer os exames a preços menores do que ele me cobra. Então, é como se o pintor for na loja de tinta, eles vão vender a tinta para ele por $1 um galão. Mas quando um cliente vai chegar, ele vai cobrar $10 para o cliente, porque o pintor ali tem um desconto. Não sei se fez sentido. Então é como um desconto que o laboratório dá para os médicos e a gente repassa isso aos pacientes. E o mesmo acontece com raio-x, por exemplo. Existem muitos locais aqui que fazem raio-x e cobram por esses exames. Isso custa mais ou menos $60. Um eletro é 45, uma tomografia é 100. Mas se eu for lá com o meu plano de saúde e eu colocar meu cartão do seguro lá, eles podem cobrar o seguro $300 pelo mesmo raio-x, ou dos 2.000 pela ressonância magnética. Então é mais barato não usar o plano de saúde aqui nos Estados Unidos. Muitas pessoas não sabem disso. E eu não sabia disso até eu começar a aprender mais sobre esse tipo de prática. E aí eu decidi, de todas as formas que eu já tentei, essa é a que faz mais sentido para mim. E essa que é mais proveitosa para mim. Isso é muito interessante, porque nós não temos nada aqui parecido com isso no Brasil. Então, para os pacientes que têm o SUS, isso, SUS. Os pacientes que têm o SUS já cobre todos os exames? Sim, a maioria deles. Claro que existem alguns testes muito caros que não são cobertos pelo SUS. Não são cobertos com tanta rapidez, ou somente se a doença realmente exigir e se os médicos exigirem, por exemplo, uma ressonância ou uma tomografia. Mas os exames básicos a gente consegue fazer dentro do SUS, dentro do programa. E nós temos alguns pacientes que pagam pelo serviço. Às vezes é muito caro para eles também, mas nós temos um tratamento particular que é como se fosse um plano de saúde, que é bem semelhante ao primeiro que a senhora explicou. Depende do plano que o paciente paga. Então, se você for jovem, eles precisam pagar menos. Doutora Lia, eu iria perguntar porque nós temos algumas coisas que a gente só consegue fazer por meio do sistema público de saúde, como transplantes ou vacinas. E eu queria saber como é que isso funciona nos Estados Unidos. Se você prescrever uma vacina, o paciente tem que pagar por ela ou ele tem que ter um plano de saúde? Como é que funciona? Essa é uma ótima pergunta. Se eles tiverem o seguro, o plano de saúde vai cobrir. Se eles tiverem vamos dizer, se eles tiver menos de 18 anos, vai ser pediátrico. Então, o plano de saúde vai cobrir tudo que as crianças precisam. Então, nós temos Medicaid para os baixa renda, que vão cobrir isso também, mas também temos crianças que não têm nenhum plano de saúde. Então, porque a família ganha muito dinheiro para o seguro do governo, mas a família não está conseguindo comprar outras coisas. Então, para eles, para essas crianças, tem o departamento de saúde, o governo ainda vai dar as vacinas às crianças, porque eles querem que as crianças sejam vacinadas. Mas o problema é mais tarde, quando você se torna adulto e você quer talvez ter mais outras vacinas diferentes, ou talvez você precisa de uma vacina de meningite, por exemplo. Então, isso não é coberto e você normalmente tem que pagar por ela, do seu bolso. Não é tão comum, porque não existem tantas vacinas desse período. Então, às vezes, eu tenho que ser bem criativo, eu tenho que ligar ao Ministério da Saúde e ligar para a farmácia e descobrir. Algumas delas são baratas, então algumas são $60, a técnicos, por exemplo, então é mais fácil as pessoas terem acesso. Mas a de pneumonia e da hepatite são um pouco mais caras. Então essa é uma área que tem um gap muito grande. Então é difícil, então a pessoa tem que decidir. Elas vão pagar por essa vacina ou simplesmente não vão tomar essa vacina. Mas para as crianças de 18 anos para baixo, a gente consegue as vacinas gratuitamente para elas. Bem interessante. Outra coisa que eu queria perguntar é sobre o gerenciamento de pacientes crônicos, a administração de pacientes crônicos. Na sua prática, como profissional particular, porque nós estamos numa equipe com um enfermeiro, uma enfermeira, mas a senhora não trabalha com enfermagem, então como é que você faz essa gestão de pacientes crônicos? A gente chama aqui de tratamento longitudinal. Bom, como é que a gente faz? O que é diferente na minha prática, a diferença é bem pequena, porque como é uma prática paga, então o que eu não expliquei antes, talvez é o outro lado, que é uma prática de associação. Então as pacientes que se associam e pagam uma tarifa por mês, essa é a única tarifa que elas vão me pagar. Então quando elas vêm se consultar, elas não vão pagar pela consulta e nem vão pagar por outra coisa. Então minha prática tem mais ou menos 200 pessoas e está fechada, está lotada. Então não tem tantas pessoas assim. E a taxa de associação por mês é mais ou menos $85 e $100 por mês que eles pagam. Então, em outra prática, você teria dos 2.000 pacientes, mas como só tem 200, eu posso fazer as coisas sozinhas e meu sistema de prontuário vai me ajudar com isso. Então, eu posso colocar tudo que eu preciso aí, eu posso me lembrar daqui a 2 ou 3 anos para saber o que eles têm, para ter algum acompanhamento, ou se eu vou fazer uma colonoscopia, para quem que eu vou ter que encaminhá-los. Então, isso facilita para mim, porque eu faço tudo. Você ficaria surpreso. Você traz o paciente, você conversa com eles, eu nem sei. Então, você está conversando com eles enquanto você está verificando. Então, eu descobri que você pode fazer muito mais do que você pode, mas é difícil quando você tem 2.000 pacientes. Você precisa ter uma equipe para isso, porque é impossível. Fez sentido? Sim. Nós temos esse número aqui de 3.000 pessoas para cada médico da família, quando a gente está num local bom para trabalhar. Porque a realidade do Brasil é bem diferente de qualquer lugar. Então, existem lugares que é muito difícil trabalhar, a população é muito grande. Então, é muito difícil fazer esse acompanhamento, como a senhora disse. Eu tenho uma pergunta que uma aluna fez, que é sobre o acompanhamento de pacientes com diabetes. Mas eu acho que é bem semelhante ao que a senhora explicou no sistema que você que pode lembrar dos exames que o paciente fez e das necessidades desse paciente. Mas uma outra pergunta que a aluna fez é sobre os medicamentos, porque aqui nós temos os remédios os gratuitos, que são parte do sistema de saúde também. Nós temos os medicamentos gratuitamente. Tem alguma coisa semelhante aí nos Estados Unidos? Bom, antes de responder essa pergunta, eu ouvi que nos Estados Unidos, para o tratamento social com pacientes com diabetes é um pouco mais caro. Por exemplo, se eles tiverem um plano de saúde, mesmo que eles sejam mais jovens, muitas das vezes eles não podem pagar. Então eu queria saber se isso é verdade ou não. A diabetes, logicamente, como nós sabemos, é uma doença muito difícil, principalmente se tiver insulina envolvida. Se não tiver insulina envolvida, ou alguns dos medicamentos novos, aí é bem mais viável. Mas quando você precisa da insulina e de coisas, de medicamentos mais novos, é um pouco mais complicado. Então, para responder a pergunta... Bom, eu esqueci da pergunta. Bom, fala de novo a pergunta. Os medicamentos gratuitos. Bom, essa é uma outra coisa que foi nova para mim quando eu abri meu consultório. Porque nessa prática, nós temos a licença para comprar esses medicamentos e fornecer aos nossos pacientes. Então, eu uso uma empresa online e eu encomendo os medicamentos e consigo por baixo preço, menor do que as farmácias conseguem. Então, Novastatina, ou por $4 por 3 meses. Então, ou talvez outros medicamentos por $10 para 9 meses de suprimento. Então, quando os pacientes precisam, eu só encomendo e eu coloco para que eles tenham acesso. Então, claro que eu cobro 10%, tem aquelas taxas de cartão de crédito, mas eles ainda conseguem com um preço melhor do que se forem comprar na farmácia. Na farmácia nós temos alguns programas dos Estados Unidos que são programas de desconto. Isso surgiu, acho que nos últimos 10 anos. Então, se eu quiser saber o preço de um medicamento, eu posso colocar e vai me dizer o preço em todas as farmácias daquele medicamento e vai me dizer qual é o preço à vista. Depois o paciente pode pegar um cupom, mesmo que eles não tenham plano de saúde, eles podem usar o plano de saúde se o seguro for pagar pelos medicamentos, ou podem fazer esse preço à vista, o que for mais acessível para eles. Eu não sei se tem outros medicamentos gratuitos, eu acho que não. Eu acho que não tem nenhum deles que é gratuito. Você tem que usar algum sistema dele, Medicaid, Medicare ou plano de saúde, mas você consegue ter acesso a eles por um preço menor do que o genérico, para alguns deles. Mas não é 100% dos medicamentos, sempre tem alguns que vão ser mais difíceis. Por exemplo, inaladores de esteroides são bem mais caros. Estou pensando em outras aqui. Tem alguns outros que são muito mais difíceis e alguns mais complicados. Insulina também é muito cara para os diabéticos, mas outras coisas para as principais doenças crônicas a gente consegue encontrar algo que seja mais acessível para os pacientes. Fez sentido? Sim. Eu acho que isso responde tudo. Eu acho que as perguntas mais importantes que eu pensei em fazer, que eu já fiz, gostaria agora de perguntar aos alunos ou às alunas, se vocês tiverem mais perguntas, o que vocês acham? Eu tenho uma pergunta. E na pandemia? Na pandemia de Covid, alguma coisa mudou em relação à vacina? O governo está pagando por alguma coisa ou não? Essa é uma pergunta interessante. A pandemia mudou a forma como nós fornecemos o tratamento, o atendimento médico, porque as pessoas ficaram em casa, então tudo era virtual. Então nas minhas consultas era o mesmo, porque os pacientes já estavam fazendo telemedicina até antes da pandemia comigo. Porque é mais fácil, eles não precisam sair do trabalho, eu fico mais confortável, a gente pode resolver tudo. Mas lembrem-se que meus pacientes pertencem a mim como se fosse uma academia, né? Então, outros consultórios, se o paciente não vai se consultar em você, você não vai cobrar nada deles. Então, se você não vai ao médico pessoalmente, o médico não pode cobrar. Então, quando tudo fechou, como meu marido, por exemplo, é cirurgião oftalmológico, então ninguém estava indo se consultar com ele. Então, ninguém estava ganhando dinheiro, porque nenhum paciente estava ali. Então, rapidamente, a maioria dos consultórios tiveram que abrir essa telemedicina, porque a gente tinha que continuar atendendo os pacientes. Talvez você não pode fazer tudo virtualmente, mas o que você consegue fazer por telemedicina, você consegue. Então isso aumentou muito, e com as testagens, inicialmente, estou tentando pensar como é que era aqui. No início, o teste de Covid começou com $50 para fazer o PCR, que não é barato, mas depois de um certo tempo subiu para $100. E esse é o preço que vocês têm que pagar se quiser fazer um teste PCR nos Estados Unidos. E depois eles estavam fazendo gratuitamente, então se você quisesse viajar, você iria à farmácia. Agora não existe mais teste de grátis. Então uma das coisas que eu acho que as pessoas estão aproveitando é o preço dos testes. Porque uma coisa é se o teste é $5 ou $10, mas $100 é muito dinheiro. Você vai levar sua família para fora do país, ou de 4 pessoas, e você tem que pagar $400 para fazer o teste de Covid em todo mundo, então não é mais gratuito, né? O medicamento é gratuito. Mas eu acho que não é muito justo. Eu acho que tudo tinha que ser gratuito. A vacina grátis, o medicamento e o teste. Mas é uma crise de saúde pública. Como é que acontece no Brasil? Aqui nós temos esse problema também do preço do teste e acesso limitado aos testes, o que é um problema, porque é uma doença muito transmissível. Então, quanto mais as pessoas testarem, melhor. Mas nós temos esse problema aqui, porque é muito caro se você for pagar pelos testes, mas nós podemos fazer gratuitamente se você for no sistema do SUS. Então você pode ser atendido no SUS. Mas nós temos as vacinas aqui gratuitamente, mas levou muito tempo para as vacinas chegarem aqui. Então foi uma questão de ansiedade para todos nós aqui. Nós ainda estamos muito atrasados no tratamento, porque nós não temos ainda alguns medicamentos. Recentemente nós tivemos o Remdesivir, mas era muito caro, acho que era R$5.000, que a maioria da população não tem esse dinheiro, não tem condições de pagar, e não é gratuito. E o SUS não cobre. Mas agora já tem outros medicamentos aceitos. Eu acho que vão ser gratuitos para o uso nos hospitais. O SUS vai cobrir, mas ainda não está aqui. Então é um momento muito difícil para todos nós. A telemedicina é algo que realmente cresceu muito durante a pandemia. E as teleaulas também, não só a telemedicina, qualquer profissão transformou em tele alguma coisa, então isso está acontecendo muito aqui no Brasil. Meu Deus. Bom, eu não falei com vocês sobre o processo aqui, mas uma coisa que eu devo dizer a vocês, eu sei que vocês querem saber o que acontece se vocês vierem para cá, mas basicamente o meu entendimento é, se você se formou na escola de medicina, na faculdade de medicina do Brasil, você vier para os Estados Unidos, você precisa ter uma residência antes de poder de começar a trabalhar. Então você tem que se candidatar para fazer o seu treinamento. Mesmo que você já tenha feito, eu acho que você tem que fazer novamente aqui. Você pode trazer o seu diploma, mas se você vai ser um médico de família, para ser cirurgião, você tem que passar por essa residência. Então você tem que se candidatar, entrar e depois que se formar na residência, aí sim você vai poder trabalhar. Isso também ocorre com os alunos que se formam aqui. Você não pode trabalhar com médico de família antes de fazer a residência. Então, provavelmente a coisa mais importante para vocês saberem é que é isso que vocês vão precisar ao chegar nos Estados Unidos. Você tem que procurar um programa de residência para depois tomar todas as medidas após isso. Eu sei que a gente não teve muita chance de falar isso, mas eu acho que é isso que vocês queriam saber também. Nós temos tempo para mais uma pergunta.
Meninos, querem perguntar?
Yes. Here in Brazil, when the
Bom,
system...
aqui no Brasil, o sistema não paga pelos exames, a pessoa vai ao tribunal e para conseguir isso. E nos Estados Unidos, existem também a mesma lógica? Quais exames? Bom, quando você não recebe os medicamentos no Brasil, se o medicamento for muito caro e o sistema público não cobre, você pode ir à justiça, você pode processar o governo e pedir que o governo dê o medicamento. Então, por exemplo, nós temos um tratamento novo de câncer que as pessoas estão tentando ter acesso, então eles estão pedindo aos advogados que entrem com o processo. Eu não sei se eles vão conseguir ou não, mas a maioria das vezes eles conseguem o medicamento por liminar. Então, se tiver alguma opção, bom, eu queria que isso existisse aqui, mas não na parte jurídica, mas às vezes existem alguns julgamentos, e julgamentos, claro que você pode ou não conseguir o medicamento, mas existem muitos programas de assistência a medicamentos para medicamentos novos, que a farmácia é um negócio muito grande aqui nos Estados Unidos, né? A área farmacêutica. Nós podemos fazer uma palestra toda só sobre a área farmacêutica aqui nos Estados Unidos, mas só para resumir, eu acho que quando alguma coisa é nova, que tem muito benefício, normalmente existem alguns programas. É um pouco difícil, claro que você tem que se candidatar, muitas pessoas, tem muita burocracia, mas para alguns deles você consegue receber de acordo com o tratamento do paciente. Se o paciente não pode pagar, ou se eles tiverem um plano de saúde, e às vezes o plano de saúde não quer pagar até que o paciente tenha feito um processo. A gente tomou isso aqui, mas não funcionou. A gente tomou isso aqui por 2 semanas e não funcionou. Então, aí sim você vai conseguir ter acesso ao medicamento. Mas legalmente, não. É mais só trabalhando com os governos, com as empresas de seguro. É assim que você pode ter outras opções. Mas é frustrante, realmente. É um processo longo. Eu acho que são todas as perguntas que a gente tinha no chat também. Eu queria agradecer muito, foi um prazer tê-la senhora aqui. E ter a oportunidade de compartilhar informações e conhecimentos e saber como você trabalha nos Estados Unidos e como nós trabalhamos aqui no Brasil. Foi muito bom para todos os alunos e para todos que estão participando. Então, muito obrigada. Muito obrigado. Esperando que a senhora esteja aqui na próxima edição. Nossa, muito obrigada por me convidarem. É impossível dizer quanto eu gostei aqui, simplesmente me conectar aqui com vocês. E ouvindo aqui a doutora Patrícia, aqui de uma colega com diferentes gerações e diferentes países, mas com práticas semelhantes que a gente encontra. Então, eu estou no Instagram, doutora Alia, eu sou bem acessível, você pode mandar mensagens no direct para mim, são assim que as pessoas me encontraram. Eu quero saber o que vocês estão fazendo também, então compartilhem comigo, me dizem o que vocês estão fazendo com os estudos de vocês e tudo isso. Então, por favor, fiquem em contato comigo e muito obrigado novamente por me receberem. Muito obrigada, doutora Alia, por sua paciência, por suas informações também, que foram muito interessantes, porque apesar de ser bem diferente, de países diferentes e diferentes sistemas de saúde, eu acho que para nós, médicos, a prática é praticamente a mesma. As dificuldades e os desafios são quase os mesmos. Então, muito obrigada. Obrigado, meus alunos. Estou muito orgulhosa de vocês nesse evento que vocês estão organizando. Muito obrigado. Ok, muito obrigado, doutora.
Our next speaker is Dr. Alexa Het. Born and raised in Winnipeg, Manitoba, Canada. Undergraduate degree in Psychology and Biology from University of Manitoba. Doctor of Optometry with distinction from the University of Waterloo. Currently practicing in Toronto, Canada, and has clinical interest in primary care, dry eye disease, and myopia control. Enjoy educating her patients and the community about the importance of eye health through social media. Her other interests include traveling, trying out new workout classes and new recipes, and spending time with family and friends.
1 second. So introduce myself before I share my screen or as I share it?
You can share your screen and then just do the rest.
Let me just exit out of my email. Okay. How do I share my screen?
It's on the bottom. It's like a green stuff.
There, got it. Is that okay? Yeah, okay. Okay, so hi, I'm Dr. Alexa Hecht. I'm from Winnipeg, Canada originally. I'm now living in Toronto, Ontario, Canada, and I'm an optometrist. I graduated in 2021, so I've only been practicing for about 7 months. And my presentation is about transitioning from a student to a doctor. So let's get started. So here is kind of an outline of what I'll be discussing today. So I'm going to go through why I chose optometry, my undergrad experience and how I got to optometry, applied to different schools in the States and in Canada, and then transitioning into graduating and getting licensed and how to find a job, and then a little bit about social media. Okay, so I kind of just went through this, but I did my undergrad in psychology and biology, and then I went to University of Waterloo in Canada for my optometry degree. And I'm currently practicing at actually 3 locations right now, so that needs to be updated, but that's recent. So, okay, so why did I choose optometry? So in Canada, optometry is a 4-year program with an optional 1-year residency. Optometry offers great work-life balance, so I wanted the opportunity to be able to, enjoy my weekends and not have to be on call. And then also there's a lot of different fields within optometry. So you can work in different practice settings, such as private practice, corporate practice. If you don't want to see patients, you can do research. And then you can also specialize further within optometry. Also, one of the main reasons I chose optometry is because there's opportunity to one day open your own business. Similar to dentistry, you can own your own private practice, and that opportunity was exciting to me. So just a little bit about my undergraduate studies. So after high school, I did a 3-year degree in the Faculty of Science, and I majored in psychology, minored in biology. In order to get into optometry school, there's a lot of prerequisites you need to do. So I kind of listed them here, but you need all your basic sciences, and then you need some more specific ones such as, statistics and English and ethics. and you do need to be taking a full course load. So that includes 5 courses per semester, and you need at least 3 years minimum. So I did a 3-year. A lot of people do 4 years. I just applied after 3 years in hopes I would get in, and it kind of worked out for me. Sorry, one second. Air, can you quiet that down? Okay, sorry. And then you need a minimum GPA of 75%. So it is quite competitive to get in. You also do need a lot of extracurricular activities. So I participated in a group called Let's Talk Science, where we led students through different science activities. I always tell people, find something that interests you and really kind of stick to it instead of doing a bunch of different things, kind of, half in, half out. So when it comes to applying to optometry schools, there are 23 schools in the States and 2 schools in Canada. One of the other schools in Canada is in Montreal, Quebec. So you do have to speak French, which I don't. So that limited me to 1 option in Canada. There's a bunch of things to consider. So, location. Do you want to live close to home? Do you want to kind of go away from home and then come back? Tuition. So in the States, it's going to cost a lot more than in Canada it would for me. But of course, if you're coming from the States and you want to go to Canada, it's going to cost you more too. Also class sizes. So my class size was 90, and in the States, I believe some of the class sizes can reach up to 200. What are the facilities looking like? So what does it look like? What does the clinic look like within the school? And something else to consider is NBO pass rates, which is the exam you take at the end of your program. It's actually a 3-part exam, and different schools will kind of talk to you about their pass rates and how many students pass each year. And then also what you need to apply. So there's an application process that happens online. You also need to write the OAT, which is the entrance exam into optometry. And that's kind of like your MCAT, pretty similar topics and subjects. And then also you need to submit your transcript and 2 letters of recommendation, shadowing experience, and then usually there's a short essay. So this is me on interview day. This is in Boston. So I applied to 3 schools in the States and then just Waterloo in Canada. So I did 4 total interviews. Usually an interview day is a tour of the school, and then you talk to a faculty member for about 45 minutes, and you have a student panel discussion and lunch. So it's usually like the morning, maybe a bit into the afternoon. And overall, some interview tips. So understanding the career is really important, knowing where the career is going to head and what the future holds for the career. And then also practicing with friends or recording yourself. So I practiced with someone who was actually trying to get into dentistry, and we kind of went through questions and scenarios, and we actually graded each other, which was really helpful. And then also, look at yourself in the mirror, record yourself and listen back. I know for me, even hearing myself speak now, I say too much. So just little tweaks like that. And then also researching the school, knowing how that program differentiates itself from the other optometry programs, because there are so many. And also have questions prepared for the faculty. And also, of course, know your strengths and weaknesses and just be yourself. They interview so many people throughout the day, and you want to stand out. So just be authentic. So optometry school is a 4-year program, like I said. There's lots of social aspects, so it's lots of fun, as well as lots of labs and courses that you're in. So we have clubs, leadership roles available, after school talks with industry leaders, conferences, social programs. Some of them are a little cheesy, but like an eyeball ice cream, which is our Halloween event. And then you also have a big sister, big brother kind of companion to help you through the 4 years. And It is a full course load. So like I said, the first 3 years, you're basically in classes the whole time, and you are kind of going in and out of clinic, but you're not seeing patients on your own. And then finally, in your third year, you actually do start to see patients with a supervisor. And then your last year is all patient-based. So you're going to 3 different clinical externships, and you are kind of seeing your own patients. Of course, you still have a supervisor, but it's more like an internship kind of role. At the end, there is an optional master's and PhD program that you can do. I guess you start at the beginning of the program. It's just more of an accelerated program, and it's a little more work, of course. And then, like I said, there's that optional residency, which is only 1 year after. And some of the specialties I have listed below. So if you're interested in something throughout school and you want to kind of specialize in it, you have an opportunity to do so. So this is kind of a day in the life through school. So like I said, you're in class or labs all day, 8 a.m. to 12 p.m. And then you have lunch, and then you have classes and labs again about till 5 p.m. And then usually after school, there's some sort of event. So like a talk, dinner, you either stay or you don't. And then you study and kind of relax afterwards. So here's how to get through an intensive program, just some tips. So find time for yourself, whether that be, working out, exercising, going for a walk, talking to family and friends back home, cooking a meal. It's important to, stay nourished, and also plan it in advance. So don't cram. I know that's a very common thing to tell people, but it is really important not to. This is kind of information that you're going to need in your career. So it's better not to just cram and forget. It's better to actually learn the material. Keep up on your notes and make a study schedule. And then also having a support system is super important, especially within the program. Find friends that have similar study styles as you do, so you can study together and quiz each other. And also, I would say join groups and clubs, outside of the classroom. So I always like to put this in presentations because I like to look back. So this is when I got my acceptance to Waterloo, and it was a really exciting day. And just like advice to your younger self, everything works out, whether you get in the first time or the fourth time. Those 4 years, you're going to grow. So don't get too hard on yourself. And then, there's no wrong path. So I guess if you start thinking you're going to go into optometry school or med school, and you realize you don't want to do that anymore, then it's never too late to change paths. Be confident in who you are. So I know I kind of touch on this later, but imposter syndrome is a big thing within these hard intensive programs. So just remember, you got there for a reason. You're smart too, and you're going to be surrounded with lots of other smart individuals. So never doubt yourself. And then also never forget to ask questions if you have them. Your profs are your friends, and they're going to be your colleagues in the future. So, reach out to them, become their friends, and never be too afraid to ask questions in class. So this is something else that's pretty cool about the optometry program, is there's international VOSH trips, which are, helping students and children and adults in underdeveloped parts of the country. And I actually got to go to Ukraine, which is really cool and also really sad right now. But, this is probably one of the last trips that will happen there, for now at least. And we got to help children in a camp. They were all orphaned children, and lots of them, had never worn glasses before, but really needed the glasses. So being able to provide them with, their first pair of glasses and the gift of sight was incredible and something I'll never forget. So I've listed some of the other sites that we traveled to. And then I know some of my colleagues did, go to Mexico and Jamaica, and they had just an amazing experience as well. So this is, if you are given the opportunity to do any sort of mission trip, I highly recommend it. It was probably the highlight of my optometry school. All right, so after, your 3 years in school, you have to start thinking about boards. And boards are 3 parts in optometry school. So you have 2 written and then 1 practical. So you take part 1 in March of 3rd year, and it's about 370 multiple choice questions, and it's your basic science. And then part 2 is more applied, so diagnosis, management, that kind of thing. And then part 3 is you're demonstrating your skills on live actors or patients. And yeah, so you start preparing for those early on. It's also just a review of everything you've already learned, but you kind of have to go back and relearn a few things. So then on to graduating and getting licensed. So you do graduate with a doctor of optometry, usually in May or June of your 4th year. And when I was looking after graduating, there are different state requirements based on what state you want to practice in. So for us, we have to do like a jurisprudence exam to practice in Ontario. And then you also have to register with college, and you get your degree and your registration confirmation. So you need all of that before you can start practicing. So I graduated in June. I didn't actually start practicing until end of August. So it takes a bit to do that. So I recommend doing that right away as soon as you can. So on to some tips for finding a job. So, look at your network. I met so many amazing, professors and colleagues within optometry school. So I actually went back and reached out to a lot of those people, and they helped me out. And if, they knew someone who was hiring, they put me in touch with them. Look on like your state website. There's always job postings. And then also, I would say just call around to clinics in your area. Explain why you are different. You just graduate, you're a new grad. A lot of people are hesitant to hire you, but the newest technology. Explain how you're valuable to their practice and how you're eager to learn and you want a mentor. I actually ended up reaching out to one of my now bosses on Instagram, and I said, I live super close to your clinic. Are you hiring? And he was like, I actually am. Come in. And we interviewed. Actually, we just chatted, and he was like, okay, I think you'd be a perfect fit. And he hired me. So you never know what can happen. Also, I think it's important to find, the right fit. You want someone as your boss who's going to mentor you and who you respect and they respect you. So don't settle. It's easy to accept the first job you're offered, but kind of continue to look around unless it's 100% what you want. You're going to want to get along with everyone in the workspace. So now on to kind of what my day looks like as an optometrist. So I usually don't start till around 10, which is so nice. So I get my mornings. I'm a huge morning person. So for me, I still wake up at like 7:30 or 8. And, I like work out or read or research or do whatever I have to do before I start seeing patients for that day. And then I usually leave my house around 9:30 and get to the office at around 10. And then I see patients in the morning, finish up charts, my lunch. I said record TikToks because that's what I'm into these days. Shameless plug there. And then also I finish seeing patients in the afternoon, usually around 5:30. And then I finish my charts and I head home for the day, cook dinner, watch TV, hang out. So it's really nice kind of transitioning from school to kind of working because you don't have to come home and study, which is amazing. And then also, I just added this in here just because I think it is important to have a social media presence, whether you're small or big. I actually got this opportunity through social media. So I've gotten a lot of panel discussions from my Instagram. I'm more new to TikTok, but I actually prefer TikTok because I think it's more fun. But, I'm going to Orlando as a transition ambassador in June. I got to travel through different, conferences and like I said, I've been given different job opportunities. So you're given so many different opportunities. Sorry, 1 second. And then also it increases your patient base, right? So I've had patients the other day. I actually had a patient who said to me, I found you on TikTok and, you looked like a good doctor, which was the coolest moment for me because it's so crazy to me that that's how social media works these days. But, even if you're not that into doing social media, just start and who cares what other people think about you? And then also one of the main reasons I actually started doing social media was because I wanted to educate the public. Optometry is not the most, well-known thing. And I wanted to educate people on why it's so important to get eye exams and eye health and eye conditions. And then also, like I said, just have fun with it, be yourself. It's all fun and games for me. So, it's nice that you get the perks of having opportunities, but I started it just for fun. And then here are some specialties in optometry. So like I said, you can specialize, but you don't need to specialize in order to do these things. So dry eye and eye aesthetics is a kind of newer specialty within the profession, but it's growing rapidly. So at one of my clinics, we kind of do specialize in dry eye, and I'm really interested in this. So intense pulse light therapy and radio frequency are newer things. They were adapted from dermatology, and they've actually found that these kind of procedures help with dry eye, as well as give you a little bit of lift under the eye. And then some clinics are kind of going into injectables and low light level therapy to help with dryness. So this is a growing market, and I think it's going to be really big soon. Another specialty is contact lenses, so hard contact lenses, myopia control, helping patients with keratoconus, which is an eye condition. And then you can also specialize in peds and vision therapy. So that kind of focuses on eye alignment and binocular vision. And here are some of my goals for the future. So eventually I do want to own my own private practice. Like I said, I'm really into dry eye, so that would be really cool to have a subspecialty in that. And then I'm always learning. I'm always kind of researching. So continue to grow and learn. And of course, go with the flow because you never know what's going to happen. So just kind of keeping your options open and never kind of being stagnant. And yeah, that's the end of my presentation. Like I said, if you guys have any questions, you can ask me now. And hopefully that was somewhat helpful. I mean, I know a lot of you aren't in optometry school, but even this can be applied to medical school or dentistry or whatever intensive program you're in. So yeah, thank you.
Thank you so much, Doctor. I think that was great. It was really amazing.
I realized I was when I did it, when I rehearsed it, it was 40 minutes, but it was only 30 minutes now.
Hi, Dr. Alexa. Can you hear me?
Yeah, you guys hear me?
Yes. Thank you for the lecture. It was amazing. And thank you for being here to answer the questions. Can we start?
Yeah.
So the first question is, what is the difference between an optometrist and an ophthalmologist?
Okay, so ophthalmology in Canada and the States, you have to go through medical school. So you do 4 years of medical school. Most programs are 4 years, and then you do your residency, and then you do usually a fellowship. So you specialize after medical school in ophthalmology. An ophthalmologist can perform surgery, so they can do cataract surgery, retinal surgery, whereas we aren't able to do that. So optometry is a 4-year program. So I'm basically like the primary care doctor of eyes. So I can prescribe medications and glasses prescriptions. I can detect diseases, manage some ocular diseases, and then if there's something I can't manage, then I refer to ophthalmology.
Okay, and next question is, why did you choose not to do an optional year of residency?
Yeah, that's a good question. I think for me personally, I had been in school for 7 years and I just was a little burnt out. I was like ready to start working and start kind of building my practice. And there was not a specific area that I was super keen on specializing in, like that they had a residency program for. So I just figured I would start practicing and see what I like and what I enjoy, and then go from there. So yeah, I would say majority of people don't do a residency. It's more so for people who either don't want to practice right away, or they definitely want to specialize in something.
Right, nice. And next question is, what was the most difficult part of your transition from student to doctor?
I would say seeing patients like on my own, because the previous year I was, you have your supervisor to kind of fall back on. And that's not to say like I have people who I can ask questions with and talk with, and people who are mentoring me. But you just feel like, you don't want to bother people all the time, which is stupid. You should bother people, but you're like just on your own, and so you have to figure things out yourself. And I think it's scary, but I think it's also what pushes you to be a good clinician.
Oh, okay, thanks. And the next question, and the last one, is what pathologies do you see most in your routine as an optometrist?
I would say I've seen a lot of like diabetes in the back of the eye, and lots of like hypertension. Like the other day, I had a patient, and you can tell if you look at the blood vessels in the back of the eye, you can see if they're like tortuous and wavy. That's an indication of hypertension. So I had like a 41-year-old patient who had very tortuous vessels, and we did a blood pressure reading in office, and sure enough, he has hypertension. So, just certain things like that where people would never know. Also, like cataracts are very common. Yeah, and then there's always the one-offs where there's random eye conditions that I see all the time. Some of them I'm still learning. So, yeah, diabetes is probably the most common.
Okay, and Dr. Alexa, thank you very much for your presence here and accepting our invitation. And we hope to see you again next opportunity. Bye-bye.
Thank you, guys.
Thank you so much, Dr. Alexa. It's a pleasure to have you here.
Bye-bye.
Nice to meet you.
Hi guys, my name is Roberta. I'd like to thank you all for watching the Congress today, and I'd like to thank the speakers for the lectures.